And so today I would like to talk to you about our efforts to actually develop new biosensors in order to investigate how the cell cycle clock ticks. So biological oscillators are actually ubiquitous in nature, and they come on all kind of time and length scales, from the pacemaker cells in your heart, which make your heart beat about once per second. Mine may be a little bit faster right now. To year-long predator-prey ecosystems, which you can find out in nature. And while all these systems look very different on the first glance, they actually all have in common that they have a negative feedback at their core. And in fact, every biological oscillator described so far has a negative feedback. So it's really a prerequisite of oscillations you can find in nature. So as my title already suggests, actually my favorite biological oscillator is the cell cycle, which you can see here beautifully in action. So these are the early embryonic divisions of the African clawed frog, Xenopus laevis. And I hope you can appreciate how highly synchronous, very fast, almost clock-like these divisions are established. However, even more fascinating than actually watching this movie is the fact that we can take the eggs from the female frogs and we can chemically activate them, and then we can pack them very tightly into a test tube, and then we can get these cytoplasmic fractions which still retain the full capacity of biochemical oscillations and the cell cycle clock. And Alessandro already did a really nice job of introducing this earlier. And you don't have to take my word for this. So we can do these experiments where we actually take now the cytoplasmic extract. So these are crushed frog eggs, don't forget this. And we can supplement sperm chromatin. As already said before, this enables actually the nuclear envelope to fully reestablish. And then we also usually add mCherry with a nuclear localization signal, which actually enables us to visualize this fully import-competent nucleus in the cell. And then we actually take a fluorinated surfactant oil. We do a very scientific step of vortexing for exactly 8 seconds, and we get these water and oil emulgent droplets, which we can then look at under the microscope. And so that's what I'm going to show you here. So again, these are not eggs. These are protein extracts, but you can already see many of them are carrying a nucleus, which is this bright dot in the middle, highlighted through the nuclear localization signal marked with mCherry. And so if you now look at this movie, I hope you can appreciate that, like in the full egg, these droplets can undergo several rounds of cell cycles, including fully breaking down the nuclear envelope, the nuclear localization signal with a fluorescent protein diffuses, and then it fully rebuilds the nuclear envelope. And so, if you look very closely at these movies, you can also appreciate that actually the area of the nucleus becomes bigger and bigger, arguing that DNA replication is also still fully functioning, as Alessandro has told us this morning. So, as with every biological oscillator, also this oscillator has a negative feedback at its core. And I will quickly tell you what we know about this. So, as the textbook will tell you, there are 3 important steps to this negative feedback. So you have the synthesis of cyclin B. And so cyclin B is the main activator of the cyclin-dependent kinase 1. Once cyclin B is synthesized and binds to the cyclin-dependent kinase 1, or CDK1, it activates the kinase. And then the active cyclin B CDK kinase complex can phosphorylate a large E3 ubiquitin ligase, the anaphase promoting complex. And upon activation through phosphorylation, the anaphase promoting complex then fully ubiquitinates again cyclin B, and thereby targeting it for proteasomal degradation, thereby inactivating the cyclin-dependent kinase again. And so this together forms a negative feedback. Many people have shown that this is what's at the core of the early embryonic cell divisions. And you could maybe ask, why do we still care about it? There was a Nobel Prize more than 20 years ago. Is there actually anything to find out? Obviously, I wouldn't be standing here if I wouldn't think that there are still interesting things to discover. And so my lab is really interested in, first of all, understanding how the necessary time delays and nonlinearities, which are also absolutely essential to make this negative feedback an oscillator, are actually established on the molecular level. Particularly also what the counteracting phosphatases, which have been neglected for a very long time, are doing in the system. And then also, if we actually understand the system so well as we think we do, can we actually synthetically build the cell cycle oscillator from scratch? However, for all these questions, one of the things we really need is we need to be able to actually measure all these enzymatic activities, ideally at the same time. And so in the past, this has been very tedious. I actually show you here data from my postdoc, where I took one of these cycling extracts, and I took a sample every 2 minutes for like 90 minutes. And then I took these samples and I measured the enzymatic activities or the phosphorylation state of certain protein one by one. That was a lot of fun. You get very beautiful measurements, right? You see like how cyclin B gets synthesized and degraded. You see how the cyclin-dependent kinase gets activated and inactivated. You see how the anaphase promoting complex, or at least one of the subunits, gets phosphorylated and dephosphorylated. And you can also appreciate how the counteracting phosphatase actually acts. But so try to motivate your PhD students to do something like this. It's going to be tough. So luckily, the people in my lab are smarter than this. And so the first part of what I'm going to talk about today is actually work from Sanna Weimar, a very talented undergraduate student in the lab. And so she really wanted to find a better method to visualize the different enzymatic activities in these cycling extracts. And we particularly focus on the activity of the cyclin-dependent kinase 1. So how has this been done in the past? We already have heard about Fred earlier today. So generally, many of these biosensors, especially if they want to sense kinase activity, are built on Fred. And they come in different designs, but all of them pretty much have this as a principle. So you have your acceptor and your donor, fluorescent protein, and then you have a short peptide sequence, which carries the consensus to the kinase you are interested in. And then they have a phosphopeptide binding domain, which upon phosphorylation, the phosphopeptide binding domain is going to bind to the phosphorylated phosphopeptide, bring the donor and the acceptor fluorophore together, and you get increased Fred ratio. For the cyclin-dependent kinase, this has first been done actually here in Cambridge in 2012. But by now, we have seen many different designs for many different organisms, which work very nicely. However, there are some problems with this. These sensors usually occupy a rather large spectral space. This means if you want to multiplex it with measuring many of these enzymatic activities at the same time, you might run out of fluorescent proteins to use. And they also require rather specialized microscopes, although compared to some of the other microscopes people are using here, they are maybe not so special. But nevertheless, we wanted to design a new biosensor. And so in order to do so, we focused on these single fluorophore kinase sensors, which had been shown in the past to work for other kinases. And they are all based on a circularly permuted GFP. So maybe you know how GFP generally looks like. This is a spiral, and it has an N and a C terminus, which are generally rather far away. However, circularly permuted GFP is kind of, so the beta strands have been swapped in a way that now the N and the C terminus are much closer together. And more importantly, that now the movement or conformational changes in the N and C terminus have a direct impact on the emission intensity of the fluorophore. And so we designed, based kind of on the general design of the Fred sensors, a biosensor which looked like this. Again, we have our phosphorylatable peptide, and then we have a phosphopeptide binding domain, which in our case is actually this WW domain of pin 1. And so the idea is that upon phosphorylation of this peptide. Now the WW domain associates, and we get increased fluorescence, but we only have to measure a single wavelength to do so. So Sanne went on, she purified the protein. She also purified the non-phosphorylatable version by just mutating the threonine, which normally becomes phosphorylated, to alanine. And we could also nicely show that the excitation spectrum exactly behaves like we would think it does with this double peak, which is usual for circular permuted GFPs. And also the emission looked really nice. And so we can now go back to our system where we can visualize kind of these cell cycles in these droplets and just add our biosensor. And we also, in the meantime, actually developed a nice a computational pipeline to actually segment and track these droplets. And so this is what I'm going to show you here. So here you can again see these droplets. You will again see the nucleus actually appearing and disappearing over time. And then on the right, you will see time tracks for our biosensor and also for the nuclear fluorescence. And so I show you 3 different droplets here. And I hope you can appreciate that we get these beautiful oscillations in the activity of the cyclin-dependent kinase in the different droplets, which very nicely follow also when the nucleus is formed and broken down again. Importantly, this has nothing to do with actually the presence of the nucleus. So we always get some droplets which don't have any nucleus because the DNA actually is distributed with a Poisson distribution in these droplets. So even if we have a droplet which doesn't have any nucleus and therefore doesn't show these oscillations in the nuclear intensity, we still get these oscillations. So we can also show that if we now use the non-phosphorylatable mutant, in these cases, we still get obviously these oscillations in the nuclear signal, but completely a flat line for the biosensor, arguing that this is really specific to the phosphorylation of the specific peptide. We got really excited about this, and we thought, like, now we put a lot of different peptides in there, and we can maybe see also different peptide specificities for the kinase or so. Unfortunately, it doesn't seem so simple, so we plugged in a couple of other peptides, and none of them actually gave us any signal. So it's not so easy to build these biosensors. So this is kind of a summary of the first part. So we have now this biosensor based on circularly permuted GFP. And we can now measure with a single excitation and emission wavelengths CDK activity. It's also very small and freely diffusible in the nucleus and in the cytoplasm. That was always a big drawback of some of the other sensors, which are so big that they actually don't get imported into the nucleus. And also compared to other translocation-based kinase sensors, they are actually working completely independently of the nucleus. And this will become important later. However, we were still not quite satisfied with this because what you couldn't see really is that we have, so far is that we have significant photobleaching. So we actually have to kind of like massage this data quite a bit to get these oscillations out of them. We were limited in the spectral range. So these circular permutated fluorescent proteins work really well in the green range, not so much if you want to go into yellow and red. And they are still quite noisy. So another very talented undergraduate in my lab, Aina Ventura-Porker, actually wanted to ask, can she do better? She's actually really excited about the frogs, but like, it doesn't look here like it. I think she was really afraid to drop the frog. Anyway, so she wanted to ask whether she can actually do even better than this. And so for that, we focused on another design for these single fluorophore sensors, and this is actually based on a circularly permuted HaloTag. So HaloTag has already been talked about a bit this morning. So it's this haloalkane dehalogenase, which is mutated in a way that it actually stops halfway through the enzymatic cycle. And so it allows you to covalently bind a fluorophore chloralkane directly to the halo enzyme. And so with this, you can actually now pick whatever color. So we also like the Janelia dyes. And you can pick kind of your favorite Janelia dye. However, like the circularly permuted GFP, also the circularly permuted HaloTag has this nice characterization that when it's bound to the dye, depending on the conformational change of the N-terminus and the C-terminus, it switches between the lactone and the Zwitter ion form, and only the Zwitter ion form is actually fluorescent. So again, we could now have a system where upon phosphorylation, we get an increased fluorescent emission. We took actually the same peptide and the same WW domain as a phosphopeptide binding domain as we had done before, and we implemented this. And to our excitement, something we had never seen actually for the circularly permuted GFP, now we could see oscillations by eye. So normally we always had to analyze it to actually really nicely see it. So I will show you this movie. And I hope you can appreciate that actually here the droplets are blinking kind of. They are becoming brighter and less bright. So that was really exciting for us, telling us that with these like better dyes, actually we can also visualize the kinase activity much better. If you don't believe me, these are the traces for some of these oscillations, and I think they look really beautiful. So now we can really start to ask actually biological questions, and we are still at the beginning with this, but I want to give you at least a flavor of what we are looking at. So the first thing we can actually do is we can not look at 2 droplets, but we can look at many of them, and we can average over them. So here I show you the different cycles for about 40 droplets each. And then we can, for example, overlay the different cycles and look at them. And I think there are 2 very striking things here. One is that the amplitude of these oscillations is really invariant, right? They look the same whether you look at cycle 1 or cycle 6, or almost the same. And this is like, I haven't normalized the data, right? This is pretty much detrended raw data, which I show you here. So this means this oscillator is really, really consistent in the amplitude. However, on the other hand, if you look at how long the period is, and you can kind of see this a little bit by where this dip and the recovery happens here, you can actually see that the cell cycles become longer and longer. And we can also actually, if we think about this in a biological manner, we can have embryonic cell cycles which are really short, right? But we can also have somatic cell cycles which are 24 hours. And also there we think actually that the amplitude of the oscillations is very similar. So we can also look at this here, right? Here again, I show you the first 3 cycles, and we can see that the period significantly lengthens. So one thing we thought is like, so maybe this has something to do with the DNA, right? Maybe somehow with accumulating DNA with every cycle, maybe this is why the cell cycles actually lengthens. But already I mentioned before that we have always these droplets with nuclei and the ones without. So if we look now and compare the cell cycle lengthening with the nucleus or without, we can actually see that there's only a very minor difference where it seems like the nucleus generally accelerates cell cycle, but the lengthening actually stays the same, independently of whether there's actually nucleus in these. We can also actually ask where does this variability come from? So we can kind of separate our oscillations in this rising phase, which is mainly interphase actually, and then the falling phase. And so if we plot over time the rising phase and the falling phase, we see that the falling phase is actually very invariant. It always is the same time, while we see that the lengthening really happens during the rising phase. And that's also very consistent with data shown actually by Sylvia Santos and others, arguing that really the mitotic timing is, or the mitotic phase is very insulated from all other phases, and it's very invariant. So we see this nicely in our data. And then one more thing I want to highlight, if we really zoom in to these cycles, we can actually appreciate that there seem to be like 4 different phases. We can see that there is this rapid increase at the beginning. Then there seems to be a kind of a phase which is more like a plateau. Then we see this rapid increase in CDK activity. This is actually normally coinciding with nuclear envelope breakdown. So this is really mitotic entry. And then we see that it decreases again. And so while some of these phases make a lot of sense, right? We have to probably synthesize cyclin here. CDK activity doesn't rise very much. And then we know that there are actually additional positive feedbacks in the system which make the system really flip into mitosis and then also go back out of mitosis. However, we found this early sharp increase in activity actually quite puzzling. And so I want to leave you with a message, which is like, although I have now also for the last 20 minutes probably talked about that this is a kinase sensor, it's clearly not a kinase sensor, but it's a kinase phosphatase ratio sensor, right? So you could actually drive the phosphorylation state of the sensor by either increasing the kinase activity or by reducing the phosphatase activity in the system. And this is actually true for all the FRET sensors out there, for example, which have been published as well. So nothing makes sense except in the light of the phosphatases. And this makes a lot of sense in our system as well. If we go back to this data I already showed you before, where we see that CDK activity dramatically rises, but we also see that the phosphatase activity at the same time actually is dramatically downregulated during mitosis. And then also we saw this very dramatic peak in phosphatase activity, much more than in kinase activity at the end of mitosis, which we think really drives dephosphorylation of mitotic substrates. And this is really what we think is actually driving the recovery from this phosphatase peak is actually what's driving here the recovery in the sensor signal. So with this, I would like to conclude. Frogs are really cool. And we have developed these new and single fluorophore CDK biosensors, which we think are really powerful and will also in the future enable us to much more multiplex these measurements with other measurements. And so obviously we have 100 different ideas where to go with this, but clearly we want to test whether actually these sensors work in other systems, especially the circularly permuted GFP-based one, which we can completely genetically encode. And obviously we are also interested whether we can measure kinase activity as nicely for other kinases. And then with the circularly permuted HaloTag-based sensor, we are really excited to actually implement this into our efforts to reconstitute the cell cycle in vitro. And with this, I would like to thank everybody involved. I highlighted Sanne and Aina before, but also Prena and Camille really contributed to this project, my whole team, and you for your attention. Thank you.
Thanks, Julia. Questions? Yeah, David.
A great talk, thank you. I'm curious about the 8-second vortex. Does it need a specific brand of vortexer or intensity of vortexing too?
So if you, if you vortex too little, the droplets become really big. If you you vortex too much, they become really small. So this is kind of the Goldilocks for us. It probably depends on the vortexer. So if you change the brand, you might be, obviously, generally, there are interesting things about having this heterogeneous kind of sizes, but it also makes a lot of trouble. They like to fuse a lot, and then obviously our measurements don't work so well. So we are really trying to go into a microfluidic system where all our droplets have the same size, and we are also much better able to control actually the size of the single droplet. But this is a quick and easy way, at least, to do it.
If I could have a quick follow-up question. Can you combine your sensors with DNA stains so you can track the amount of DNA in the droplets as the cycle progresses?
Yeah, I think so. We haven't done this, but I think other people have just used DAPI or Höchst, and you could actually look at it. You could also use recombinantly. I mean, one of the power of these systems is you can add recombinant proteins. So you could add, I don't know, histone GFP or something if you wanted, and you'd probably be able to measure that too.
Question up there.
Thanks, nice talk. I was just going to ask, over time, are you losing RNA? Is it actually the synthesis rate of cyclin B that's changing and delaying? And what do you think the shoulder is? What switches you from a shoulder into mitosis in your view?
Yeah, I don't. So the honest answer, we don't know why the cycles slow down. So other people have postulated that it's ATP. So although then I think nobody ever have succeeded by using ATP recovery systems to rescue it. So I think it's still an open question. RNA is supposed to be really stable in this system. It's actually one of the directions we want to go, is to look now at cyclin B synthesis in addition. So we would be really interested to do this. And then, so I think the shoulder and the switch come from that, in addition to cyclin B, the cyclin-dependent kinase is actually regulated by these positive and double negative feedback loops mediated by WEE1 and CDC25, which really regulate this switch. Interestingly, we have very recently done experiments where we inhibit WEE1 and the switch still looks very much the same, arguing again a bit like along the lines of what my postdoc work also argued, is that the phosphatases are really important, and maybe the whole switch actually runs via the phosphatases much more than via the kinase itself. So we're really interested in looking at
I
this.
suppose a lot of that work suggests the nucleus plays a role, but yours works without DNA, just the same.
Yeah.
So there's no.
So we can rule out that the nucleus has a major role in this. Yeah.
Perhaps I can ask a question. So you talked about changes in the periodicity, but the amplitude stayed exactly the same. So what's the biological significance of that, do you think?
So I think the significance is that, so from a biological point of view, the significance is that probably you want to ensure that you have a defined amount of kinase activity to get your cell or your droplet into mitosis at that point. And probably you don't want to have too much, and you definitely don't want to have too little, otherwise they get stuck actually in mitosis. From a more theoretical point of view, this is kind of this phenomenon of tunability, right? So you have an oscillator which is tunable in period, but not in amplitude. Again, positive feedback has been implicated in being like underlying this phenomenon, but we are also really interested in looking into this more.
Okay. Any questions online? No? Okay, if there are no further questions then, we can draw this session to a close. I'd like to thank Julia and the other speakers again.